Hi everyone, Kevin here, back with video number 5, the last video in Module 2, Topic 4. Let's get going. This is an example of a glycosylated compound found in grapes. And the molecule, the aroma molecule that is glycosylated is linalool over here. Linalool is the compound that gives much of the flavor to Fruit Loops cereal. So if you eat Fruit Loops, or you did as a kid, you know a little bit what linalool tastes like. It's a very common terpene, and we'll talk about the class of aroma molecules called terpenes. It's a very common terpene found in certain grape varieties. And so linalool can be a free aglycone, in other words, a free aroma molecule in grapes, but it can also be glycosylated to sugars. So the question here is, how might a glycosidic link form between this linalool and this sugar here? Well, we know, we've seen this a lot right now. We've seen that the lone pairs on that hydroxyl group on linalool could attack that anomeric carbon in the sugar right there and make a glycosidic bond. So we've got a hemiacetal in the sugar right now, and we could have one more attack by an alcohol to form an acetal. So remember, acetal is the reaction of an aldehyde or a ketone with two alcohols, and this is an example of one alcohol having already attacked the carbonyl group, the aldehyde in this case with glucose, and that was the intramolecular attack by the hydroxyl on carbon 5. Remember that from a couple videos ago. On this slide, we're looking at the second alcohol, i.e. the hydroxyl group on linalool, attacking then and forming, changing the hemiacetal into an acetal, forming the acetal. All right, hopefully that is clear. Be sure you are pretty well versed with the formation of these glycosidic links. And there is the product, linalool linked to the glucose. Hydrolysis can break glycosidic linkages. So we said in yeast, oftentimes the yeast break bonds, conjugated links, covalent bonds between aroma molecules and sugars or amino acids. And that can be a chemical hydrolysis or an enzymatic hydrolysis. And so in this case, we're actually showing that molecule that we looked at on the prior page, linalool, bound to glucose. And there's an enzyme in bacteria called glucosidase that will break that linkage between the two. It's a hydrolysis, so water is added. And we'll look at enzymatic mechanisms of doing this in a second. But you're back to then the aglycone, linalool, smelling wonderfully like Fruit Loops and separated from the sugar to which linalool was previously conjugated. All right, so this is hydrolysis, but it can be a chemical or an enzymatic reaction. Here's the chemical reaction. Here's a little bit of detail. So you'd be in an acidic environment. It's acid catalyzed hydrolysis, and that is fine in wine because we've got plenty of acid around. And so the proton is going to get picked up by this oxygen here. It's going to lead to this protonation here with a nice positive charge on that group, and then we will get removal, bond breaking, as, let me change colors here, as these electrons go back to the oxygen, we get the bond breakage, and the alcohol, which is shown here, leaves, and you're left behind with the glucose, and shown here is a resonance structure of the glucose, and that then picks up a water. You lose a proton, you rearrange, and you end up with a hydroxyl group back here at this position on the anomeric carbon. The squiggly line indicates that you could have the hydroxyl either above or below the ring, depending on how the mechanism worked. So that's it. That's basically basic chemical hydrolysis of an enzyme of a glycosidic bond. So I'm going to circle again that glycosidic bond here in green. It's right there. We protonate that. That allows that promotes the bond breaking, the breaking of that glycosidic link between the carbon and the oxygen, and the alcohol leaving. Now, I said that hydrolysis can be chemical or enzymatic, so here is an example of enzymatic hydrolysis. So, first we have the sugar here bound to some other molecule, that's the OR here, right? So that is the conjugation. There's a glycosidic linkage between the sugar and that OR group. Or the glycosidic linkage, I suppose you could say, technically includes the oxygen and it's linked to the R group. All right, so to start the process, we have our substrate in the active site of an enzyme. If you haven't had biochemistry, you'll talk more about this in class, and we'll talk about it here as well. But you can think of enzymes as proteins. Usually they're proteins. And that protein is, the structure of that protein is such that you create a little pocket in the enzyme in which your substrate can fit. In that pocket, there are amino acids lining the inside of that pocket that promote certain reactions to happen. So enzymes catalyze chemical reactions. And those amino acids that line that active site of the enzyme are often there and in exactly the right position to promote certain chemical reactions of the substrate which has entered the pocket of the enzyme, the active site of the enzyme. So if you can imagine here, we show two amino acid side chains, and I'm going to circle these in blue. So one of them is here, and the other one is down here. And both of these are connected to the enzyme. So the enzyme would be a long protein that would wind around here, connecting these two amino acid residues and containing usually hundreds of other amino acids that make up your big protein, which is your enzyme. Okay, and I'm just drawing an outline as if we knew what this looked like. We really don't, but what you do know are the two amino acids that are active in this chemical and catalyzing this chemical reaction are in these two positions shown here. All right, so position, the enzyme or amino acid 1 and amino acid 2, I will label them. Now both of these are similar. They both have carboxyl groups sitting in the active site. And you see this one carboxyl group here is protonated. It's going to serve as an acid. In this case, it's going to donate a proton to make this reaction go. The other amino acid that also has a carboxyl group here sticking into the active site of the enzyme is going to serve as our nucleophile. It's deprotonated already. So to catalyze the reaction, then you have the substrate here in green. It comes into the active site of the enzyme. It's positioned such that it can pick up a proton by this arrow here that I'm pointing out. That black arrow shows the picking up of the proton. That would be the first step. And then you'd have attack by the nucleophile on this carbon here. That's the anomeric carbon. And you'd attack that, and that would break the bond, and this OR group would leave. All right? And we'd end up with a sugar and the OR group separate. So that's an enzymatic reaction. So there's an enzyme involved. Enzymes catalyze reactions. Enzymes are other molecules that are little molecular machines, really. In a sense, they catalyze reaction because the active site of the enzyme is lined with amino acid residues in the right positions to catalyze certain reactions. And here we're showing 2 amino acid residues in the active site of the enzyme. Both of those residues have carboxylic acid side chains that stick into the active site and catalyze this reaction. So that's how it goes. So hydrolysis can be chemical, just acid catalyzed, or it can be enzymatic. And we find both of those in wine. We certainly have chemical hydrolysis, especially in the bottle as the wine is aging, because at that point there's no live yeast, there are very few enzymes, but there's lots of acid. So you have acid catalyzed hydrolysis, and acid catalyzed reactions of a lot of kinds that change the flavor of the wine over time. One reason wine lovers love wine is because it evolves over time and often gets better over years and years, even 20, 30, sometimes at 100 years if a wine will last that long. It's pretty amazing stuff. So here shows the products of this enzymatic reaction. Again, it shows the alcohol here leaving the ROH. The OR group picks up an H. And then it has the sugar remaining over here. All right. And there we go. There's the final, after the sugars left the active site. The enzyme is back to where it was originally, and that's the nature of catalysts. They catalyze reactions without being consumed. So the enzyme is back to where it was at the very start. You can buy enzymes to add to wine, and here's one. Scott Labs is in Petaluma. It's one of the big suppliers of the wine industry. You can buy from them enzymes that will help liberate flavor compounds. So you see this enzyme mix has 2 enzymes in it, a pectinase that breaks down pectin in grape cell walls. We'll talk more about pectin later, and beta-glycosidase, which is one of those enzymes that frees aroma molecules from sugars. So you can buy these and add them to your juice. Here are more examples of glycosylated aroma compounds. So have a look at these. You don't need to know all the compounds, but I hope you'll be able to recognize what the compounds look like when they are liberated from the sugars. Some of the sugars are monosaccharides, some are disaccharides, some can be even longer. But in each case, hopefully you can pick out the aroma compound pretty easily. And that's it. Thanks for watching the last video in topic 4. This is actually video number 5, and we will see you in the next module.